thank you so much for inviting me. This really, thank you, Nia, and all your whole staff for being so welcoming and for hosting this terrific conference. I've really been enjoying it and been very impressed with the results that you've been getting from the thin brain study. One of the things I think about it at this stage in my career is what's going on with the young investigators, and I feel so joyful and so in some ways envious of you folks out there who are just in the stage of embarking on your careers. I think that the tools that we have now, the large data sets that we're developing are just amazing, and I think that you will make such wonderful discoveries over the course of your career, and I'm excited to hear about what you're all going to accomplish. So I'm going to talk about the type and timing of maltreatment and the effects on the developing brain. And so the first big question is, what does maltreatment do to the brain? And I started off doing this research approximately 30 years ago, and at that point in time, we were basically thinking that that stress is bad for the brain, and stress is particularly bad for the developing brain. It was about that time that Doug Bremner had published a book, Does Stress Damage the Brain? And Bruce McLean was doing wonderful work showing the effects of glucocorticoids on the hippocampus. And it was, you thought about it as, early life stress was going to damage the brain. And and then basically started doing the research with that thought in mind. But over the course of time, it struck me that does this really make sense? That through all of the eons of evolution, that the mammalian brain has not been selected to be relatively resistant to the effects of early life stress, since that's been such a significant part of our ancestry. And so, in thinking about that while working on an article in Scientific American, it struck me that doesn't really make sense at all, that the brain's being damaged. And what the logical alternative is that exposure to early stress generates molecular and neurobiological effects that alter neurodevelopment in an adaptive way that prepares the brain to survive and reproduce in what is predicting to be a potentially malevolent world. Now, again, it was pointed out yesterday in talking about the evolutionary theory, that what's really important is reproductive success. And it doesn't matter if this is going to produce problems like shortened lifespan or a great array of psychopathology. What's really key is that you get to reproduce and to pass on the genes. And that the brain is not damaged, it's very selectively and specifically modified. So the first principle is that maltreatment in general does not result in non-specific brain damage, but rather produces specific phenotypic modifications, which we propose may foster survival and reproductive success, but may also be highly maladaptive at other stages of development or in other circumstances. And so what might be a beneficial phenotypic adaptation? So that if you're being raised in a world in which there's a lot of risk of harm, what major phenotypic adaptation may be to be better able to detect and respond to threats. And a lot of the brain circuitry is designed to detect and respond to threats in the environment. And this is our latest circuit diagram on the neurobiology of threat detection and response. There'll be a quiz later. But basically, sensory information comes in, and all the sensory systems except for the olfactory system are relayed through the thalamus, and there's a pathway through the thalamus that's relatively direct and can rapidly go to the amygdala and can produce responses like alterations in interoception, activation of the hypothalamic pituitary adrenal axis, defensive reactions and defensive actions, through its connections, and these all can happen, relatively quickly, 30 to 40 milliseconds, below the level of conscious awareness. And so This is very important in having a very initial survival response. On the other hand, information also goes up through a high road to primary sensory cortical regions, and then to secondary cortical regions, and this brings it into our perceptual awareness, the threat. And then there's a very interesting information loop that takes place between the sensory systems and this part of the ventral lateral prefrontal cortex, particularly inferior frontal gyrus, which sort of sets the threshold for information coming through and getting to the amygdala in terms of threat. And this can modify the amygdala's response. And this region here is also very involved in language. It's part of Broca's area. And it's maybe to bring it into and in connection with the language system brings the threat and the feeling of things like fear into our conscious being. The amygdala itself doesn't produce fear. It's basically bring it into our consciousness that produces the sense of fear. And then there's projections to the ventral medial prefrontal cortex, which can inhibit amygdala response. There's projections to the dorsal anterior cingulate, which can activate and amplify amygdala response. And also medial prefrontal cortex plays some role in balancing these factors. And we'll focus on some of the key components, the thalamus, the visual cortex, the fusiform gyrus, which is involved in face recognition, amygdala, hippocampus, this pars triangularis, and these prefrontal regions. And so looking first at the amygdala, which plays this central role in fear detection and response, the amygdala is a key limbic structure critically involved in encoding of implicit emotional memories and detecting and responding to salient stimuli, such as facial expressions and potential threats. The structure and functional abnormalities in the amygdala have been detected in a wide array of psychiatric disorders, social phobias, specific phobias, unipolar and bipolar depression, addiction, autism, borderline personality disorder, and schizophrenia. And we know from animal studies that exposure to threats produces persistent neuronal hypertrophy and symptoms of anxiety. It particularly targets the pyramidal cells in the amygdala and causes more dendritic arborization and branching and more synaptic boutons. And that this is very, very enduring. It doesn't significantly reverse with time. If you look at the response of the amygdala to threat in the scanner, you'll see amygdala activation, and it's particularly activated on the right side to threat. In fact, the laterality is so strong that individuals who are looking at animals and looking, say, at fear conditioning in rats only look at the right amygdala. If you look in this case, this is work by Delanowski, looking at the childhood trauma questionnaire score. The higher your childhood trauma questionnaire score, the greater on average is your amygdala activation to threat. And what we've been concerned about is the idea of sensitive period. If the brain is going to be altered, programmed by stress, it's probably going to depend on the stage of development in which the stress occurs. And that what we've postulated is that brain regions that are going through rapid developmental change may be particularly targeted by stress. It also turns out that as the brain regions develop, there's often an excess of excitatory synaptic information, and at a certain stage in development, the inhibitory processes come into balance, and it's during that period of time when the brain regions become very open to modification by environmental factors, and then molecular breaks come in that reduce the plasticity. But it's not that the brain can't be plastic, it's actually being inhibited from being plastic, and then certain things like the psychedelic drugs can open up these plasticity effects and make the brain much more plastic again, so you can potentially modify things and have things like therapy work much more efficiently by opening up another window of plasticity. And so we looked at this with the amygdala. We did this in conjunction with Carlin Lyons-Ruth, who's a attachment researcher at Harvard Medical School. And Carlin has a sample of individuals that she studied for attachment behavior when they were infants, using stranger paradigm, and then she's continued to follow them for about 30 years, and we had got funding to bring in a representative subsample of her population for neuroimaging, and it was a small sample, only 18 adults, and many of them had mothers who were approach avoidant, leading to disruptive attachment. We compared them to healthy controls, and what we found was that both the right and left amygdala were slightly larger, significantly, compared to the controls, and that on the right side, it correlated with the degree of exposure to childhood maltreatment, where on the left side, it didn't. And looking at it specifically, it looked like exposure to maltreatment at 10 or 11 years of age was particularly associated with an enhancement in amygdala volume on the right side, but not on the left side. But we found in terms of the left side that that disorganized attachment behavior, maternally disrupted communication, and overall attachment risk correlated. This was measured at 18 months of age, and it correlated with amygdala volume 30 years later. So it looks like the right side may be affected by various forms of maltreatment, and the left by problems with attachment. And so we figured that there are two critical developmental threats that take place. One is the profound threat of rejection or neglect, and that maybe the left amygdala is specialized for perceiving this kind of threat to the attachment bond, and then there's other kinds of abuse and injury that maybe the right amygdala is more specialized for. And this makes sense if you think about the specialized function of the right and left hemisphere. The left hemisphere is specialized for the perception and expression of emotions that lead to approach behaviors. That includes positive emotions, and it also includes anger. Whereas the right amygdala is specialized for the perception and expression of emotions that lead to withdrawal behavior. So if you're detecting a threat to the attachment bonds, withdrawal would not be the appropriate solution, and then approaching might be a much better solution. Whereas if you're detecting other kinds of threat, withdrawal might be a better response. Now, that's dealing with the amygdala from a structural standpoint. We've also looked at the amygdala from a functional standpoint. And it's interesting, the literature has found that there are some disorders that are associated with an enhanced amygdala response to threat, such as anxiety disorders, and there are other disorders that are associated with a blunted amygdala response to threat, such as in substance use disorders, and or other types of inappropriate social behavior. And maltreatment's a major risk factor for both types of disorders, both for internalizing and externalizing disorders. So we wanted to see if there were factors that are associated with an enhanced amygdala response and forms of maltreatment that are associated with an enhanced amygdala response and others that are associated with a blunted amygdala response. So we wanted to identify this type of timing, and we had 202 participants, and to look at this and to look at the sensitive periods, we developed an instrument called the Maltreatment and Abuse Chronological Exposure Scale. Angela Paraga came from Germany for a year to help work on this scale. And it's sort of a version of the ACE. We initially thought about it as a modified ACE, but and we eliminated some factors from the ACE. There was a nice talk on that yesterday. We eliminated factors such as history of mental illness or history of substance use, because that confounds experience with potential genetic risk. We eliminated separation or divorce because that could mean very, very different things to very different people. And it's more important to actually look at the behaviors that they're experiencing rather than the events. We also eliminated incarceration for the same reasons. We added in peer emotional and peer physical abuse as very potent forms of maltreatment. The ACE is also limited to witnessing abuse to mothers. We expanded that to witnessing interparental violence, and also included witnessing violence to siblings, which is an underexplored but very potent form of adversity. And so we had 10 types of maltreatment. We also found out that emotional abuse separated out in terms of factors into a verbal and nonverbal component of emotional abuse. So we ended up having 10 factors, and on the scale, we inquire about exposure to each of these 52 odd items that lead to these measures. across each year of childhood. So we wind up getting a developmental course, a retrospective developmental course. And we can see that different types of maltreatment have their own different time courses. Things like emotional neglect and physical neglect wax and wane only a small amount. Other things like parental physical maltreatment peak at a very early age. Things like nonverbal and verbal emotional abuse by parents tend to peak later in childhood. There are also substantial gender differences, such that sexual abuse will show a much greater effect on females, particularly after age 7, and males experience more severe physical bullying than females. So this enabled us to potentially look at type and timing of maltreatment. One of the big problems in doing this is that there's a lot of collinearity in the data set between maltreatment exposures at adjacent ages and between different types of maltreatment at the same age. And to really pull that apart, we looked at different machine learning techniques and found that random forest regression with conditional inference trees served as the best means of detecting the underlying predictive factors in this data set, even in the face of significant collinearity. And Elliptigon Khan in the lab has been working on looking at the relationship between type and timing of maltreatment in psychopathology. And Jinjin Ju, who came from China to complete his PhD thesis, has been looking at this in terms of risk on brain function. And so in this study, we had individuals doing the Hariri emotional matching task, face matching task, and they're basically exposed to fearful and angry faces, which are forms of threat, and in contrast to neutral faces, and in contrast to simple geometric shapes. And we looked to see how much there was amygdala activation, in this case in the comparing response to negative versus neutral faces. And this is the analysis which shows you the effect of emotional neglect, nonverbal emotional abuse, peer emotional, peer physical, parental physical neglect, parental verbal abuse, sexual abuse, witnessing interparental violence, and witnessing violence to SIBS across each year of childhood. And this shows you the importance of each factor as a risk factor. You can see how important nonverbal emotional abuse is at around 8 or 9 years of age. This blue line is the influence of the multiple types of maltreatment. The red line is the importance of overall severity of maltreatment. The green line is the importance of duration of maltreatment. And you see that more important than multiplicity are things like exposure to specific types of maltreatment at specific ages. And that's true for many factors, that it's not really the multiplicity. Multiplicity seems like a big risk factor because it increases the probability that you'll experience one of the critical determinants of the actual response. And when we look at those, we see that some of the facts of maltreatment lead to a blunted response, and some of the types of maltreatment lead to enhanced response. And that particularly things like emotional abuse at age 14 and parental and peer verbal abuse at age 17 and 18 leads to an enhanced response, whereas peer physical at 6, witnessing interpersonal violence at 7 and 9, physical neglect at 9, nonverbal emotional abuse at age 9, peer physical at 11 lead to a blunted response. And we separated out individuals who experienced only those factors, one or more of these factors that led to enhanced response. These individuals who experienced one or more of these types of maltreatment that led to a blunted response, but not the later factors, and those who experienced none of these factors. And you can see in response to the negative versus neutral stimuli that in those who experience none of those, you get a somewhat greater response to the negative versus neutral. In those that have had these, you get this much greater response to the negative versus neutral face. Whereas in those individuals who experienced the early abuse only, you actually get a reversed response, where they're actually responding more to the neutral face than they're responding to the negative face. So it's really a big difference in terms of whether you've had this exposure basically pre-pubertally or during the post-pubertal period. This makes sense from the standpoint that with exposure to maltreatment here, it may be good to have an enhanced amygdala response to threat, and that you can produce a more robust fight or flight response, or a most robust HPA axis response. In this case, the blunted response, or the reversed response, is interesting. We're thinking that it has a blunted response to stress because if being exposed at these ages, you can't mount an effective fight or flight response to threat. And that what may be happening is if you're experiencing, parents who are both nurturing but at times, abusive, that if you had too much of a fight or flight response, it may actually interfere with the attachment bond. And that it may make sense to actually blunt the response so that you're not having too much of that, which is maybe adaptive in that period of time, but may set the stage for further problems down the road. In terms of the clinical implications of this, looking at risk for various psychiatric disorders, unfortunately there weren't that many subjects in the early exposure group only. We had a lot with non-exposure, we had a lot with late exposure. You can see that things like major depression were substantially increased by late exposure, that generalized anxiety was increased, risk for specific phobias was increased, substance abuse was increased, eating disorders increased, personality disorders were increased by this late exposure. It actually may be that the early exposure reduced risk for major depression, reduced risk for general anxiety disorder, also was not effective in enhancing substance use or eating, but did lead to an increase in OCD and did lead to an increase in any personality disorder. So that's basically the amygdala in a nutshell. Moving to the hippocampus, similarly the hippocampus is a key limbic structure critically involved in the formation and retrieval of explicit memories, including autobiographical memories. And this is also affected in a great number of psychiatric disorders, including PTSD, major depression, schizophrenia, bipolar disorder, drug addiction, and borderline. And as Christine Heim pointed out yesterday in her wonderful keynote, that there's been, seminal work that was done showing that that it wasn't really depression that's affecting the hippocampus, it's really the maltreatment experience that's affecting the hippocampus. And I think that's a key finding in the field. Stress in many ways exerts the opposite effect on the hippocampus that it does on the amygdala. It also targets the pyramidal cells and causes reduction in the dendritic arborization and reduction in the number of synaptic boutons. It also targets the dentate gyrus and suppresses neurogenesis in the hippocampus, and so it leads to generally a smaller hippocampal volume. Studying hippocampal volume has been Carl Anderson in the lab and Kyoko Ohashi, and we found that the simplest way of pulling the data to understand the effects on the hippocampus was to specifically look at the effects of abuse versus the effects of neglect, and we found that there was a very, very prominent gender difference. In males, it was neglect basically during the first 7 years that was associated with a reduction in hippocampal volume, not abuse, whereas in females, they weren't affected by neglect, but they were affected by abuse, particularly at 10, 11, and 15 or 16 years of age. In terms of functional response, we found that, again, teenage exposure to emotional neglect and parental and peer verbal abuse at 15, 16, 17 years of age led to an enhanced response, and that witnessing interparental violence at 9, peer emotional at 9, peer physical at 10, this 9 or 10 year age being very important, led to a blunted response in terms of the hippocampus. We've also looked at the sensory cortex, and this also overlaps nicely with Christine's work. And we had Jiwook Choi come from South Korea, was a visiting scientist, Akimi Tamada from Japan as a visiting scientist. And we basically were exploring the question, does the nature of the maltreatment matter? This is many years ago. And we recruited individuals with exposure predominantly to one specific type of maltreatment. And we looked at fiber track using diffusion tensor imaging and track-based spatial statistics. We looked at gray matter volume using voxel-based morphometry. And what we found is that individuals who had high level of exposure to parental verbal abuse had a specific alteration in gray matter volume in the auditory cortex, and they had an alteration in the integrity of 3 fiber pathways. One that was most affected was the arcuate fasciculus, which interconnects Broca and Wernicke's area as an important language pathway. And these were, a whole brain analysis where all the data is thrown into the computer, and the computer identifies what was significantly different. We didn't pre-plan this. It pulled out just the auditory cortex and this important language pathway is being affected by parental verbal abuse. In contrast, witnessing domestic violence, this is visually witnessing multiple episodes of domestic violence, affected gray matter volume in the visual cortex, particularly the primary visual cortex, and affected the integrity of one key fiber pathway, which is the inferior longitudinal fasciculus, which interconnects the visual cortex with the limbic system and determines our memory and emotional response to things that we see. So really specificity with the verbal abuse affecting the language pathway and visually witnessing domestic violence affecting visual system and visual limbic integration. And then we looked at childhood sexual abuse. We found alterations predominantly in the visual cortex also, the secondary visual cortex, and Christine, as she pointed out beautifully yesterday, found alterations in the somatosensory cortex that are responsible for sensation from the clitoris and surrounding genital area. So there's important specificity, and that in terms of sensitive period, looking at domestic violence and visual cortex, that it's predominantly around 12, 13, 14 years of age in which the visual cortex is being modified. We know from the work of Hubel and Wiesel that the visual cortex is highly plastic up until a period of around puberty, and after puberty it ceases to be so plastic. So we're getting this transition right at that juncture. The other region that hasn't been talked about much in the maltreatment literature is the thalamus. And the thalamus is this key relay station where all the sensory systems are coming in. And we've recently completed a study with Colin Lyons, with a new study, where we recruited about 150 mothers of newborns. We were able to image 92 of the mothers. A collaborator, Ellen Grant at Children's Hospital in Boston, neuroimaged about 57 of the children, and we've been looking at their brains and comparing them. But if we look at the mother's brain, we found that the volume of the right thalamus is affected by two factors. Since we only had 92 subjects, we couldn't use all 10 types of maltreatment. We broke it into 4 categories: emotional deprivation, emotional threat, physical deprivation, and physical threat. And we found that emotional threat, particularly early in development, and physical threat later in development were associated with alterations in the volume of the right thalamus. And it's particularly emotional threat at age 3 and emotional threat at age 4 that was associated with an increase in thalamic volume, whereas physical threat at 9 and physical threat at 11 were associated with a significant decrease in thalamic volume. On the left side, similar story. We get the early emotional threat leading to an increase in volume, and we have physical threat basically from age 9 to 12 leading to a reduction in thalamic volume. Now, the thalamus is a very complicated structure that consists of a lot of nuclei. We're particularly interested in the lateral geniculate nucleus, which is the relay portion for the visual system, from the eyes to the visual cortex. The medial geniculate nucleus, which is the relay center for the auditory cortex, and the ventral posterior lateral portion, which is the relay nucleus for the somatosensory system. And these are all attenuated by exposure to physical sexual threat around 9 to 11 years of age. In terms of the regions that are enhanced, it's particularly the ventral anterior magnocellular part and the central medial part, which are enhanced by this early exposure to emotional threat. This part's involved in functions related to motor control, motor planning, execution, regulation of motor tone, and sensory intermotor integration. This part is involved in emotional regulation, and dysfunction has been linked to a variety of psychiatric disorders. It's interesting, it's normal to think that bigger is better and smaller is worse, and that if you reduce the volume, it's going to make things, function more poorly. If you enhance the volume, it's going to make things function, more effectively. It's probably the fact that during brain development, you go through periods of overproduction and you go through periods of pruning. And I think that when the brain is being modified early in life, it's working off of the overproduction process and amplifying the overproduction to produce the changes, and you often get an increase in volume. Whereas with later exposure, you're working off of the pruning process, and I think you tend to get a reduction in volume. And so there may be alterations associated with both an increase in volume and a decrease in volume. We also looked at this in terms of functional effects. In this case, we looked at individuals and we exposed them to highly arousing positive or negative images versus neutral images. And normally in an fMRI study, you compare response to one type of image to the other type of image. In this case, we also had them looking at a fixation point. So we're able to compare response to a neutral image versus a fixation point, response to a positive image versus a fixation point. And what we found, which was really striking, is that that in terms of brain regions that were affected by maltreatment, that maltreatment had the biggest effect on response to the neutral image. Not the negative image, not the positive image, but the neutral image. And particularly emotional abuse and physical and sexual abuse affected response to neutral images. And across a very, very large number of brain regions, there was effect on maltreatment on processing the neutral image, less effect on the negative image, less effect on the positive image. And that what we found was that we divided the individuals into those who had a big response to the neutral image and those who had a small response to the neutral image. And that specifically increased your risk for PTSD by tenfold, having a robust brain response to the neutral image, but no other psychiatric disorder. And we believe that this is essentially a marker for hypervigilance, and that individuals who've been exposed to maltreatment may be unable to, sort of relax and not worry about, a negative scene. And these are very boring neutral images. These are pictures like an umbrella against a wall or just a simple chair. But they may be looking for hidden dangers, and I think that's a fundamental aspect of PTSD. In terms of the prefrontal cortex, there's a variety of prefrontal cortical regions affected. Same story, that we get response particularly to verbal abuse at later teenage years and emotional neglect in middle teenage years are associated with an enhanced response, and those early are exposed with a blunted response in the dorsal anterior cingulate. Similar story with the ventral medial prefrontal cortex, and similar story with the with the inferior frontal gyrus pars triangularis. That's particularly, this is part of Broca's area, and it's particularly responsive to parental verbal abuse and reduced by peer physical abuse, and you get this change. McCrory and his colleagues proposed that functional abnormalities to threat processing would act at latent vulnerabilities and increase the risk for developing anxiety disorders rather than directly producing symptoms of anxiety. And we looked to see how much these alterations in bold response were predictors of increased risk for anxiety disorder. And we found that having a 1 standard deviation increase in response to the negative bold response in the pars triangularis, the inferior frontal gyrus, increased your odds of developing a anxiety disorder by 5.5-fold. And in contrast, having an increased response in the ventral medial prefrontal cortex, which can then in turn suppress amygdala response, reduced your odds and made it 6-fold less likely that you're going to have an anxiety disorder. So you get to shape the response by altering the function of these things, which is based on the type and timing of maltreatment. So the second principle is maltreatment exerts prominent effects on key components of the threat detection and response circuit, and can lead to a blunted response with early exposure. Later childhood exposure can lead to an enhanced response. Sensory systems, which are a primary window to the external environment, are selectively modified by types of maltreatment that they convey, as Christine was pointing out yesterday, to reduce the impact of the threat during that stage of development, but then may have profound consequences later in development. Stress-susceptible brain regions appear to have windows of vulnerability when exposed to specific types of maltreatment, and associated with the most significant alterations in brain structure and function. And evidence suggests that brain regions may have two distinctly different sensitive exposure periods, which may be associated with opposite long-term effects on volume and brain function. And that's the basic story in a nutshell, and thank my staff who've been doing this research and our large number of collaborators, and thank you for paying attention.